All right, let's dive into one of the most incredible processes in all of biology, meiosis. We're going to unpack this amazing cellular journey, which is the special type of cell division that makes sexual reproduction and all the wonderful diversity of life even possible.
Okay, so here's a question for you. Have you ever stopped to think about this? You get a set of chromosomes from each of your parents, right? So why don't you have double their number? It's a really good question, and it gets right to the heart of the problem that meiosis is designed to solve.
To get our heads around this, we need to know two key terms. Most of the cells in your body are what we call diploid. We write that as 2N. It just means they have two full sets of chromosomes, one set from each parent. For us humans, that's 46 total. But the other type of cell, a haploid cell, or N, has just a single set. You can probably see the problem now. If a diploid egg and a diploid sperm got together, the baby would have four sets of chromosomes, and that just doesn't work. So nature had to figure out a way to cut that number in half first.
And that, right there, is the entire mission of meiosis. It's designed to take one of those specialized diploid cells and, through this really elegant dance of division, produce haploid cells. You know them as sperm or eggs, or, more technically, gametes. The whole point is to reduce that chromosome number by exactly half, making sure the next generation gets the right amount of genetic material.
So how does it pull this off? Well, it's a two-act play, meiosis 1 and meiosis 2. Each of these acts has the same familiar stages: prophase, metaphase, anaphase, and telophase. The whole process is basically one round of copying the DNA, followed by two rounds of cell division, one right after the other. That two-part structure is the secret to the whole thing.
Let's kick things off with meiosis 1. The big event here, the main goal, is to separate what we call homologous chromosomes. So what are those? Well, think of them as matching pairs. You get a chromosome number 1 from your mom and a chromosome number 1 from your dad. That's a homologous pair. Now, before any of this starts, each one of those chromosomes has already copied itself, making two identical sister chromatids that are stuck together. The whole point of meiosis 1 is to pull those homologous pairs apart from each other.
Okay, now we get to the real main event of meiosis 1. It's called prophase 1, and it is, without a doubt, the most action-packed part of the whole journey. It's way longer and way more complicated than the prophase you'd see in a regular cell division. So much is going on that science has actually had to break it down into even smaller steps. Yeah, prophase I is so intense, it has 5 substages. You can see their names here. Now, don't sweat memorizing them. The names are a bit of a mouthful, I know. What's really important are two critical things that happen during this whole sequence.
The first critical event is called synapsis. This is where those homologous chromosomes are matching pairs, find each other in the cell, and pair up really tightly, matching up gene for gene. This little bundle is called a bivalent, or sometimes a tetrad, because if you count them, there are 4 chromatids altogether.
And then comes the really cool part, maybe the most important event of all, crossing over. While those homologous chromosomes are all paired up, they actually swap little segments of DNA. They literally break off pieces and exchange them. This is what shuffles the genetic deck, creating brand new combinations of genes. It's a huge reason why there's so much genetic variation out there. And here's what it looks like. You can see the homologous pairs all snuggled up together during synapsis, and they're actively swapping bits and pieces of their DNA. This is the moment that guarantees the final cells are not going to be simple copies of each other. After all that action in prophase 1, these homologous pairs, now with their freshly shuffled genes, all line up together right down the middle of the cell. The key thing to see here is that they're lining up in pairs, not in a single-file line like you might expect. Next up, in anaphase I, the homologous chromosomes get pulled apart towards opposite ends of the cell. But, and this is absolutely critical, look closely. The sister chromatids that make up each chromosome are still attached to each other. We're only separating the pairs at this point, not the individual chromatids.
And that brings us to the end of meiosis 1. In telophase 1, the cell splits into 2, so what we're left with are 2 brand new haploid cells. They've got half the number of chromosomes as the cell we started with. But you'll notice each of those chromosomes is still made of 2 sister chromatids.
But we're not done yet. The whole mission was to create gametes, and those sister chromatids need to be separated. And that's the job of meiosis 2. This second division is actually much simpler, and it looks a lot like a normal cell division, a process called mitosis. Its whole job is just to separate those sister chromatids. So now we've got two cells going through the process at the same time. In metaphase 2, the chromosomes in each cell line up individually along the middle. This probably looks pretty familiar if you know about mitosis. It's just happening with half the number of chromosomes. And here we go. The final split, anaphase 2. The little structures holding the sister chromatids together finally divide, and the chromatids get pulled to opposite sides of the cell. The second they separate, we stop calling them chromatids. Each one is now considered a full chromosome in its own right. Okay, if there's one thing you take away from this whole process, let it be this difference. In anaphase 1, it's the homologous chromosomes that separate. In anaphase 2, it's the sister chromatids that separate. Nailing this distinction is really the key to understanding all of meiosis. So after all that pulling and splitting, what do we end up with? From that one single diploid cell that started this whole journey, we get a grand total of 4. And there you have it, the grand finale. The process wraps up with telophase 2, and we are left with 4 haploid daughter cells. And what's so amazing is that thanks to that crossing over way back in the beginning, every single one of these 4 cells is genetically unique, different from each other, and different from the parent cell that started it all. So, okay, we've walked through all these steps, all this complex choreography inside a tiny cell. But why? Why does any of this matter so much? What's the big picture here? Well, it all comes down to 2 huge mission-critical jobs. First, meiosis makes sure that the chromosome number for a species stays the same generation after generation. It keeps the genetic blueprint stable. And second, just as importantly, it actively creates genetic variation. It's the engine of diversity. And, this quote from our source material really just nails it. The genetic shuffling that happens during meiosis creates new combinations of traits, and those variations are the raw material that natural selection has to work with. Without meiosis constantly creating these new variations, evolution would basically grind to a halt. All right, let's do a quick recap, kind of like a cheat sheet. Meiosis I is what we call the reductional division. It reduces the chromosome number. It separates homologous chromosomes, creates variation, and turns one diploid cell into 2 haploid cells. Then meiosis 2 is the equational division. It's more like a simple equation, where it separates the sister chromatids and gives us our final 4 haploid cells.
So I'll leave you with this to think about. We've seen how this tiny cellular dance shuffles genes. If you consider that crossing over and the fact that humans have 23 pairs of chromosomes that can line up randomly, how many genetically unique children could a single couple theoretically produce? The answer is staggering. It's somewhere in the trillions. And it's a powerful reminder of the incredible engine of diversity that is meiosis.